Morning everyone, good to see several of you online. Morning Tanishka. So this is going to be part 2 of the Biology 3, Infection and Response. So part 1 we did on Monday, and looking back we went through the difference between communicable and non-communicable diseases. We looked at how a pathogen is any microorganism that causes disease. There are lots of microorganisms out there, we live with them all day, every day, we're covered with them both on and in us. And actually a lot of them we have sort of a fairly symbiotic relationship with that we not rely on, but they do help us. So for instance, the bacteria in our gut helps digest food. And there's actually some evidence now that the bacteria that we have in our gut is very dependent on the types of food we eat. And there is actually some evidence that the feedback from that bacteria dictates the foods that we want to try and eat in the future. So if we do try and change our diet, it actually takes a little while for our body to adjust and to want the new foods, because the bacteria that's actually inside our gut has to change as well. And I can give you a couple of links for that later on. We then went through and we had a look at bacteria, we looked at viruses, fungi and protists. We looked at the type of microorganisms, and we had a look at some of the diseases they actually cause. So make sure that you can go back through and you can do those. Remember I said that the example diseases that I gave you, they're the ones that you have to remember. You have to know the symptoms, you have to know potential treatments, but bearing in mind the exam board can ask you about different diseases that you haven't learnt about before, but they would expect you to apply the information that you know. So for instance, they could talk about a viral disease that you've never known about, but you should be able to use your knowledge of viruses being smaller, living inside cells, being difficult to treat, to apply that knowledge to the new disease. They will often give you the new symptoms, so they won't expect you just to miraculously know stuff. They'll give you enough information, but the background information we went through the other day is expected to be known. Now, what I want to do today is I want to have a look at the sort of body's defences against pathogens. So we're looking at actually how does the body prevent infection in the first place, and then what happens once we become infected. I am also going to give you a link. If I'm going to give you a link now, this is another YouTube channel, and the main reason I'm going to give you this link is because it's a channel that I use in school quite a bit. They are incredibly well funded and the quality of their videos is absolutely exceptional, and I can't do a video as good as they can. So what I'm going to do is put that on there, and I will also drop it into the Google Classroom later on as well, but you should be able to copy that later on. I'm not even sure how you pronounce the YouTube channel's name, but they do some absolutely amazing videos, so well worth, after this particular one, at some point over the next couple of days, go and watch that video, because they've done a brilliant one. Now, the first point of call, I suppose, when we're looking at disease, and this is absolutely relevant at the moment, is stopping the spread of disease. So in other words, we need to have a look at different ways that pathogens can be passed from one person to another, or from one object to another. Remember, we are looking at communicable or contagious diseases here. We are not looking at non-communicable, which would be dictated by lifestyle, or just by being unlucky. So we're looking at ways that we can physically pass them on. Now, we should know, because it's in the news all over at the moment, particularly with the coronavirus, that they're putting systems in place to reduce the spread. And I can come on to the coronavirus a bit more later on, because it's technically a whole topic in itself. But if you think about the general government guidelines, when we're looking at this, one is they're talking about avoiding close contact. Now, obviously, outside of the sort of coronavirus, that's going to be very difficult to do. We like gatherings, we like getting together with other people, we like the whole social element, most of you. So the whole sort of trying to keep a distance is working at the moment to try and reduce the spread, to try and allow other systems to be put in place to try and relieve the pressure on the NHS. But it's probably not viable long term. They're also looking at the use of face masks. Now, we know that actually face masks, there are lots of different qualities. We're not looking at surgical grade here, which would have quite a big effect at stopping you catching it. What it is trying to do is stop the spread of droplets that we breathe out. So we've got that method as well. There's also, and I think I've got a picture of this, that everybody should recognise, because this is not just for the coronavirus. This has been with the flu and for colds for quite a long time. We've got this idea that actually if we are coughing, if we are sneezing, then we want to try and catch it in a tissue. Or they're saying crook of the arm, whatever, but ideally in some form of tissue to stop that spread. And there's some quite interesting videos out there that I think Mythbusters and other people have done that actually talks about how far when you sneeze, those droplets can actually spread. And it's quite a long way. So it's worth having a look at that as well, and it spreads at a very high speed at that point. They're obviously looking at binning it then, because actually once that tissue's gone somewhere else, nobody else can pick it up and catch it. And then the key thing here is killing it by some form of sanitation to our hands. But I'm going to come back to that as we sort of start working our way through. Now, none of that should be new. It's even if without the coronavirus, this particular poster has been on the wall. It's been around for years. So it's something that we should know about. So let's have a look at ways that we can catch the disease. Obviously, touch. Let's go. So if we put our hands on a surface that has pathogens on there, that has microorganisms on there, some of those microorganisms are going to be transferred to us. Now, that doesn't necessarily mean we are going to become ill with that disease. Okay, morning, Olivia. It doesn't mean that we're actually going to become infected. It does mean that we've got some of those pathogens on, and there is a likelihood of us becoming infected, depending on what we do next. If we were to touch a surface, and actually we were to wash our hands well afterwards, then hopefully any of those pathogens on our skin then gets killed. So we haven't become infected. But if we then start touching our face, if we start picking up food and eating the food, if we then go and touch other people, if we touch other objects, then actually we can then pass on those pathogens. Some of them will go from us onto that other surface. So it can spread from one place to another quite quickly. Now, there are various sort of studies done, and a lot of this depends on the material. Certain materials, I think there's been studies done with copper, definitely with silver ions, then the different pathogens can't actually withstand that. So the contact and the survival time on that surface is quite low. Other contacts, some plastics, some woods, and generally other surfaces, they've had the viruses, and I've seen reports from between 3 and 7 days that particularly with coronavirus can actually stay alive for, or stay viable for, because being a virus, it cannot survive outside of cells in terms of propagation, so there is some sort of discrepancy over whether it actually classes as a living organism or not. But that's a discussion for another day. I'm going to resist to use the word debate for the moment. Okay, so we've got this idea of touch. Then we've got airborne. Is it B-O-R-E or is it B-O-U? I can never remember. Anyway, we've got airborne. So we've got this idea then that it's passed. This can be through droplets that we breathe out, which is normally the case. You don't get that many spreading on the wind. Fungal infections, you can, because fungi produce spores. So remember, this isn't just all about COVID. We're looking at other types as well. So we've got this idea of this airborne, these spores that can spread out, and they can also remain on surfaces as well. So if you look at things like athlete's feet, athlete's foot can often be spread from changing rooms, from walking around barefoot, if their hygiene isn't kept up to date. But again, it's a fairly easy one to treat. Obviously, waterborne. So we're looking at waterborne. It's a very common one. There are lots of diseases that can be spread through contaminated water. Now, the contamination could be from animals living locally. It could be from humans. So it could be from human waste, or it could be from, as in bodily waste. So we're talking about sort of fecal of urine. It could be about contaminated food, etc., that has managed to wash into the water supply. So there's lots of things that can cause waterborne diseases, and these are quite proliferant, throughout the world. I know there were reports once about there was a lot of people that, and still do, that go whitewater rafting along the river that goes to run through Grand Canyon. And one of the problems they had was, because this was often a multi-day trip with camping, a lot of the people were urinating and defecating too close to the water supply. And because of that, a lot of it then would wash into the river, and people becoming ill quite quickly if the water wasn't treated sufficiently. So in other words, the water at that point was not potable. And the term, I'll put this down here, that just means drinking quality water. So I can put that down there. Okay. So it's quite easy to contaminate water in that way. And I know now that if you are doing expeditions down rivers like that, it's not so bad with urine, but for any sort of fecal matter, tissues, etc., you're actually expected to bag it out. So you go in the bag, everything goes in there, and they actually even provide capped tubes. Everything goes in the tube afterwards, and you can get quite a big fine if you don't choose not to do it that way, because they're trying to reduce the contamination of these water sources. We don't just get it there. I mean, this can happen in the UK as well, especially in the winter. So if you get a lot of people to expedition mountaineering in the snow, the problem with the snow, particularly when it's quite deep, is you've got to get through the snow to get to the ground. So if you are going to the loo, if you're going to bury it, you've then got very hard ground as well. And there are lots of people in areas such as the Cairngorms that were choosing not to go deep enough, or they couldn't get into the ground, which means when the snow melted, you had lots of piles everywhere, which could then be washed into the water stream, and wasn't particularly good to look at. So they've been encouraging bagging out for places like that as well. So yeah, apologies if you just got up and having breakfast. Not the best of topics to be listening to today, but it's quite important. So we're not going to go through specific airborne and waterborne diseases. There's lots of information online if you want to do the research, but you need to be aware of that. Now, in terms of treating water, from a home point of view, all of our water is treated by the water treatment plants. So it'll be filtered. So normally, if we're looking in sort of industry, we would be looking at, we've got filtration, followed by some form of sterilisation. Now, that's the way, there we go. Okay, sterilisation. So that could be ultraviolet, or it could be chemical. And then what we normally do as well is we'll use some form of chlorination. Now, this is not just chlorine gas. The chemical, by the way, can often be ozone. The chlorination will be something like hypochlorite, which is similar to what they use in swimming pools, but at a much, much lower level. And this helps prevent the water becoming contaminated again. Now, if we're outside, there's other things that we can do. So we wouldn't have access to all of this. We still have filtration. So if I'm on Exped, I will generally have some form of filtration device with me. And these can range from different bags to caps that go onto the top of water bottles to actual pumps. And some of these have been proven to pretty much reduce the contamination down. They won't ever put 100% down, because just in case something gets through for legal issues, but it can be sort of 99.99% efficient. We also have chemical. So depending on where I'm going, I may back it up with, I've actually got some iodine, which I can use to put in the water. My wife absolutely hates. She won't use it at all, just because of the taste of it. But it works quite effectively. And we can also use ultraviolet. Now, I've never tried this one by myself, but you can actually buy ultraviolet light that you drop into a water bottle, and then you have to leave it for a certain amount of time. And obviously the most effective method is boiling. But bearing in mind, boiling won't remove dirt, it won't remove pathogens that are inside the dirt unless you leave it for a long period of time, and it won't remove chemicals. So what we generally are expected to use is a combination of filtration and boiling when we're doing this one. But waterborne disease is quite important, particularly if, for some of you that may want to go travelling in the future, so when you finish school, you've got your qualifications, possibly got through your A-levels as well, and you want to take some time out either before or after university or other jobs, then actually if you're going travelling, we need to be aware that waterborne disease is probably one of the most likely causes of becoming ill. Some countries don't treat their water to the same extent, and you can actually use some of this equipment, and you can actually buy it to fit onto taps. So it's worth being aware of, which is why I spent a little bit longer on waterborne diseases than possibly would do normally. Obviously food we can have. We know that contaminated food, we've spoken about salmonella before, And in a lot of cases, this is down to the food not being cooked properly. It would have been contaminated before, but in a lot of cases, there is an issue with even though it's been contaminated, if you cook it properly, that contamination should, the toxins, the bacteria, any pathogens should be broken down. Now, remember, we are talking about microorganisms, so this is not going to have any effect on chemical contamination. So chemicals into the water supply, chemicals into food, heavy metals, that sort of thing. We're not talking about that at all. We're purely talking about pathogens today. But in the same way as waterborne, both of these we're looking at ingesting. So we're looking at eating or consuming the contaminated products here. Now, we can also have vectors. So vector would be some form of pathogen that is carried on another organism. So we could be looking at mosquitoes. We could also be looking at dogs. We could be looking at cats. We could be looking at other animals. Now, we know for a fact that actually the dogs, if we get bitten by dogs, the chance of infection is huge. The bacteria on dogs' teeth is quite high. And straight away after a bite, we can become contaminated with all sorts. And actually, in a lot of people, it's the infection that's caused by the bite that does more damage than the actual bite itself, because that can be treated quite quickly. So vectors is when we actually get contaminated by by pathogens that are carried by another organism. Right, let's just move that down. So if we start looking at the sort of human defences, we've spoken about things we can do, and a lot of this comes down to cooking food. And that's always interesting when you see, so going back to the cooking for a moment, I mentioned, I think last time, it's well worth, if you get a chance, I think it's only about 15-20 quid, but you can do an online food hygiene course. And it's quite an eye-opener just to see how things work, see how we can be contaminated the types of food. And it's one of those qualifications that you can then put on your CV. So if you want any more information on any of those, let me know, and I'll post you in the direction of a link that you can use, but it's quite well worth doing. But it's when you start looking at different foods, so I quite like my meat rare, but it's still perfectly safe, because although most of the bacteria for this is going, or any pathogens, is going to be on the outside of the meat. So by cooking the outside, you kill that off, and it shouldn't have got too deep into the meat, which is why it's safe to eat beef and things rare. However, if you start thinking, there are some restaurants that do these really expensive burgers, and they're actually making, and I can't remember what they're called now, it begins with a W, I'll think of it in a minute, but they're producing these burgers where the mince is steak that's been quite coarsely ground, and they're still cooking it rare. So what you're doing is any of those pathogens that's on the outside of the burger is then getting mixed into the inside of the burger and not getting cooked. So there is a higher potential on that. Now it's quite low, or restaurants wouldn't be allowed to do it, but it's one of those things I've never quite got my head around yet. So if there's anybody out there that knows a little bit more on that, then let me know, because I'd be quite interested in how they are actually safe to eat. I have eaten them myself though. So if we start looking at the sort of first, I call this the first line of defence. So what we're looking at now is how do we stop becoming infected? So we haven't maybe cooked our food properly, or maybe we've been touching different surfaces. What are the body's natural defences that tries to stop an infection? Now, I'm going to come on to the actual immune system in a moment, because the immune system is when we're looking at white blood cells, where we're looking at other issues. So we're not talking about that for the moment. I'm going to come on to that next. This is literally barriers and methods. So the biggest barrier we have is our skin. If you think about it, it is massive. It's pretty much the largest organ in the body because it covers the whole of the skin. So this is a barrier that hopefully most pathogens won't get through. And we know that because actually we can touch contaminated surfaces, and if we wash our hands afterwards, we don't get contaminated because the pathogens on our skin gets cleaned, gets killed off, and actually we are not then infected by anything else. On that note, when we are washing, soap is actually one of the most effective, and if you use soap properly, it's more effective than the alcohol hand gel. I've seen lots of students before carrying around pots of alcohol hand gel, and that's absolutely fine. It works, and it works particularly well when you haven't got access to running water and soap, but it's not as effective as soap. And the reason why soap works so well is all organisms have this like a fatty layer around the outside. And you can actually see the effect that soap has on fats. If you put a little bit of oil on top of some water and float it, and then drop some washing up liquid or some liquid soap onto it, it starts to break it up into smaller droplets, it starts to break it down. And that's what happens with the pathogens on our skin when we wash our hands with soap. As we're washing it, and they reckon we should wash our hands for at least 20 seconds, partly to make sure the soap's in contact long enough, and partly to make sure that we've actually covered all of our hands. But what's happening is the soap itself is breaking down the fatty layer in the bacterial cells, in the fungal cells, in the viruses, and it's breaking them down so they literally fall apart and they're no longer effective again. And that's something that a lot of other chemicals don't do as effectively as just a bar of soap. So soap is one of the most effective methods. That's not just the news and everything saying, yeah, you must use soap. There's actual science behind that, and we can talk about that a bit more later on if you want to. But yeah, you can do tests on this at home, but literally forget the alcohol soap, the alcohol gel you're at home, just use soap. So we've got that as a barrier. We've also got this idea that we've got platelets in our blood which can form scabs. So when we cut ourselves, the blood starts oozing out. That does help to clean it to a point, but obviously we've got an opening straight into our body at that point, and from our body that's not good, because that is a route that infection can take hold within and get into the system. So platelets start oozing out, and those platelets will lock together and bond, and they will form a scab over the wound, and that stops us then becoming infected again. So platelets and scabs are the next sort of method when we're looking at trying to stop anything getting through that giant barrier that's covering our body. If we look at our eyes, it's I say rare. It's not that rare, but we don't generally tend to get an infection of the eyes that often. We can end up with conjunctivitis, which is quite contagious. But most pathogens, we have this sort of liquid around the eyes. Sort of the tears that when we cry, and when we're blinking lots, we get things in our eyes. We've got this liquid around it, and that has sort of antiseptic qualities, which kills off a lot of the pathogens that stops us getting an infection of the eye. Now, we know that I think it's a bacteria, I could be wrong, but I think it's a bacteria that causes conjunctivitis, isn't killed off by this, so that can take effect and that can get into the system. But generally, that's one of the methods that stops us getting infection through the eyes. If we look at the nose and we look at airways, a couple of things here. We've got the mucus that's produced, and we produce lots more of this when we have a cold. And we also have the hairs and the cilia. So if we look at the sort of hairs in our airways, we have the cilia that's produced, and the cilia help to move mucus and debris. So the mucus actually helps to trap pathogens and dust and other debris, and then the hairs on the cilia actually move it out the way. You might want to note that mucus is actually produced by a specialised cell called goblet cells, and they produce it, so they ooze it into our airways, and then hopefully that will trap any pathogens and they'll move it out of the way. And again, yes, we do go down with chest infections, with lung infections, but bearing in mind how much we actually breathe in every day, we don't get infected that often. So that's another line of defence for the body. Stomach, we have acid and we have protease enzymes, and generally this makes quite an inhospitable environment that a lot of pathogens get killed off. Again, we know it's not all of them, because actually we do end up with food poisoning. If you've had food poisoning, it's not particularly pleasant. Like I said, I think in the last video, I have had salmonella, and I felt rubbish for about a week. You get lots of vomiting, lots of diarrhea. It's not caused by the bacteria itself, but rather by the toxins that they produce. But the salmonella bacteria itself is not killed off by the stomach acid or by the enzymes, but a lot of the other ones are. Because actually when we're eating things, as I said before, we are surrounded by bacteria, by microorganisms. So even whilst we're eating, even after it's cooked, some things will be landing on the food. So we live with it and we don't become infected very often. There's not enough on there. It gets killed off by the acid, and then the rest of it gets killed off by the immune system before it can take hold. It's when we get larger numbers that we start getting issues. But that's sort of the next line of defence there. So those are the sort of main defences that try to prevent the infection. The problem is what happens once we become infected, and that's where our immune system kicks in. Like I said, I thoroughly recommend the video that I posted at the start of this, and I'll put it into the Google Classroom as well, or into the resources on my website. I thoroughly recommend watching it. They have the quality of their animations and the way they explain things is brilliant, and they've done a really good job of trying to explain how the immune system works. It starts off with them giving you an idea of how complex it is, and they bring it down to quite simple words. So yeah, it's well worth a plug, well worth having a look at that. That is the video I generally use alongside what I normally do, but when I'm teaching in lessons. So the first thing to know when we start looking at pathogens when they get in is most pathogens have antigens. So antigen is a protein or an area that is unique to that particular microorganisms. And they are all different. And our body takes some time to try and learn to recognise the particular antigens for a pathogen that might be into the system. And until it's learned to recognise it, it can't do anything about it. So we're going to go through and explain what it does. We're not going to go into too much detail in terms of how, because that's quite complex. And those of you that go on and do A-level biology, or you start looking at doing histology in the future, you'll do more of that then. But we're looking at these sort of proteins, but we're also designed for recognition. Design isn't the right word. It's not exactly like the pathogens suddenly turn around and go, oh, I'm going to put this on. This is my name. Here I am, by the way. It just means that every type of microorganism will have just different protein structures, and we call these antigens. But they're areas that our body can learn to recognise. Okay? Now, we have different types of white blood cells. For the sake of GCSE, there's two main types that you need to learn. One of them is called lymphocytes. And these are the ones that produce antibodies. So antibodies are structures that are produced by white blood cells, and they are specific to a particular type of antigen. So let me just change colour for a minute and we'll talk a little bit about antibodies. So these are specific to each antigen. Now, our body can make pretty much an unlimited number of antibodies once it has encountered an antigen. But until we've encountered it, we won't know how to make it. So once they're made, though, they have some quite major uses within the immune system. So let's just say we've got, I'm just going to draw, let's just say this is a bacteria. On that bacteria, there will probably be these little protein structures all the way around. And I'm just going to draw them really simply as an arrow. So these are going to be our antigens. Now, once the white blood cells encounter it, what they're going to do is start producing other structures called antibodies, and these antibodies can bind to the antigens. No, so the lymphocyte is a white blood cell that produces antibodies. So they learn to make them, and then they are the production factories for the antibodies. But the antibodies are a type of protein. They're not a cell in themselves. So what the antibodies do is they bind themselves to the antigen, and then they can either cause lysis, which is the breakdown. So I'm trying to make sure that we're using the correct terminology here. Lysis is when a cell basically bursts and falls apart. So it can either cause the white blood cells to break apart, or it can cause them to sort of clump together. So they join together and they end up as a bigger group of pathogens. Sorry, forgetting what I'm saying then. Group of pathogens. What they also do is they can be recognised by the other type of white blood cells, which are called phagocytes, and these are the ones that do the destruction. So they will, phagocytes, and we'll come to those next, they will start enveloping and breaking down some of the white blood cells, but they can do it much more effectively when the pathogens are all joined together, clumped together, and the fact that it can then recognise them. So the first thing we've got is these lymphocytes. They produce the antibodies. The antibodies are specific to antigens. So the antibodies bind, so we can put bind here, to the antigens, and that then causes lysis, clumping together, and allows them to be recognised by the phagocytes. So that's the first thing that happens. The next one we've got are these phagocytes. And these literally are, they're pretty much destroyers. They're the ones that go round and they call that. There's a process called phagocytosis. Ooh, cytosis. Basically what they do is they engulf any pathogens, anything foreign in our body. And then once they do that, they use enzymes to cause lysis and to start breaking them down. So anything they come across they don't recognise, they'll do this to. But if there is, if they've already been clumped together and basically tagged by antibodies, they do this much more effectively and much bigger. So it takes a little while for our body to learn to fight different pathogens once we get infected. And one of the sort of graphs that's quite common, you need to be able to explain with this. So if I just draw, oh, let's do that as a line. So what I've got is time. This is going to basically give us our rubbish. This is going to give us pretty much our level of infection. Or we could turn around, we're going to say it's like our antibodies and antigen. Is that going to still work? Give me. So what we end up with is a graph. So I've just dropped my pen in my coffee, if you want to giggle. Luckily there's no electronics in it, so I just need to dry it out before it works properly. But I can still use the mouse for a minute. So what we end up with up here, this is going to be the level of antibodies. And if we get infected, to start with, our line's going to be quite low. We're going to get infected about here. And then what we end up with is this very, very slow raise. And then it drops back down. But it doesn't drop back down to 0. So what we then end up with is a period of time. And if we ever become infected again, notice the difference. Okay, so we become reinfected here. And this is really a very, very common graph that you will get in an exam. It comes up quite a bit. And what they're trying to ask people to do is they want you to be able to explain why the graph is the shape it is. So I'll just talk through it again. The graph starts off with very low numbers, pretty much 0 of the particular antibody we need in our bloodstream. When we become infected, then those numbers gradually start to rise as our body learns to fight it. Then we're actually ill for quite a period of time. But afterwards, the levels of our antibodies don't drop back down to 0. And we keep some in our body. And these will be stored as memory cells, which means if we ever get reinfected again at some point in the future, because our body doesn't need to learn to identify and make them, we get a massive increase in antibodies really quickly, which means we don't start seeing the symptoms of that. Okay, so hopefully that makes sense. If I can get this working again. No, I'm going to have to let that dry out before it works. I always wonder, actually, I wonder if this will work. No, it needs a pen. Okay. So, what I would do is annotate this graph. I can't at the moment because I haven't actually got my pen to work currently. But annotate the graph and make sure you've got your heads around what's going on. So, this is our antibody number. We've got a period of time. We get infected. Our body learns to produce the antibodies and starts producing them. Then as the numbers go up, our body starts fighting the disease. So it's fighting this disease here. Afterwards, we have no more pathogens in the body, so the numbers drop back down. But they don't drop back down to 0. They drop back down to a level. And we've got these memory cells that recognises these pathogens. After that, if we get reinfected again, and this is the reinfection, because our body can recognise them, it knows they're there, then we get a very, very rapid increase in antibodies, which shoots up and means we don't get infected again because they can actually kill the disease before we have that level of reinfection. Okay, so it's worth just making sure you've got that annotation there. Right, going on to next. So the other thing I need to put up here that I missed, if we're looking at, and I might have to just write this in text. If we're looking at antibodies, we've also the lymphocytes also produce antitoxins. So if you remember when we looked at, hang on, there we go. If you remember when we actually looked at salmonella, we said that the symptoms of salmonella are caused by the toxins that are produced by the salmonella bacteria, not by the bacteria itself. So we need a way of getting rid of the toxins that have been produced by those pathogens. So the lymphocytes, as well as producing antibodies, can also produce antitoxins. And they are particular enzymes, particular chemicals that will break down the toxins that have been making us feel ill. So that's the sort of second use, the second function for the lymphocytes that would be worth writing down. Now, generally speaking, so this bit here, we could say, go here, hang on, this bit here, right with the mouse just for a minute, it'd be very bad. We would technically have immunity against that disease. Now, this is the area that sometimes confuses some people, because one of the things they say is, but I get a cold more than once. So what happens, and we're going to look a lot more of this when we go on to do evolution in future lessons, but what happens is if you think about the multiplication rate of pathogens, so we said that bacteria can multiply every 20 minutes, that means that every single time we're going to multiply, we have the chance of a mutation happening. Okay, so if that mutation happens, then there is a chance that we'll end up with a different type of pathogen. It may still have the same symptoms, but the one thing that's going to be different will be the antigens on the surface. And at that point, our body no longer recognises that pathogen and it can't fight it. So at that point, we've got to relearn to do it again. But for most sort of diseases, because the diseases aren't very common, once we've had them, we only get them once. So things like chickenpox, generally, and there is an issue with chickenpox because you can end up with shingles afterwards. I've got no idea what that on Tanishka. That won't be caused by anything to do with pathogens. That would be caused by something else. I'll do some digging around and see if I can find out, but I don't know any. I've not heard of that one before, but that won't be caused by an issue with pathogens. Okay. Now, this idea of immunity it's not something that was that new. So there is a scientist that you need to know, and I'm gonna have to type because my pen's not working at the moment. And we were looking at a scientist called Edward Jenner. Now, Edward Jenner started doing some research, and there's lots of videos looking at his history, so I'm not going to go into it in huge amounts of detail. But Edward Jenner started looking at smallpox. So we can put down smallpox here. And I mean, smallpox, when you look at it, was a disease that caused quite horrific deaths. It caused massive sort of spots and blistering of the skin, and lots of people were dying of smallpox. People that survived smallpox never went down with it again. But one of the things that Edward Jenner noticed is that there was a slightly different version of smallpox called cowpox that obviously the cows had, but it wasn't fatal. Now, the milkmaids back at the time, who used to milk the cows fairly regularly, lots of them went down with cowpox, and they would end up with small sort of pustules on their hands, and that's about it. But none of those ever went down with smallpox. So he came up with this theory, and he wanted to test the theory to try and find out whether there was something in being infected with cowpox that stopped you getting smallpox, or whether it was just a random observation. And as scientists, one of the things we do is we like to test things. Obviously, the health and safety back then isn't what it is now. Because while I describe what he did, I just want you to try and think about what would happen if anyone tried to do this now. So he found a young boy that had not had either cowpox or smallpox. What he did was, using a scalpel, he made a small cut on the boy's arm. He then took some of the pus from the pustules on a cow, and he rubbed the pus into the cut on the little boy, and he deliberately infected the boy with cowpox. Sure enough, the boy ended up with a cowpox, but because it's not fatal, he survived it. So the next part of his theory was to find out whether that boy did not go down with smallpox. So what Edward Jenner did is he created another cut on the boy. So he cut this boy open again, but this time he went and collected some pus from the pustules on someone who had smallpox. And again, he rubbed that pus into this boy's cut and infected him with smallpox. The boy did not develop any of the symptoms. So from that day on, that was pretty much the first form of vaccination. Now, we've developed it over the years to the point that obviously we don't need to rub pus into people's cuts, because I think most of you would probably whinge quite a lot at having that done. I mean, let's face it, lots of you whinge about getting up early. So the thought of having it being cut open, having pus rubbed in, probably wouldn't go down too well. But what we have done is we've now worked out that when we vaccinate, we inject somebody with either dead or slightly altered version of a disease, so that that person's immune system can learn to recognise the antigens, because that's the most important thing. We're trying to give somebody the antigens without giving them the disease, so the body can learn to recognise them, so the body can then learn to fight it. So that's what's happening with a vaccination. And vaccinations have meant that millions upon millions of people have survived by not becoming infected with diseases that they would normally have become infected with. And actually, when you have a look at it, the smallpox disease has pretty much been eradicated worldwide through vaccination, which means nobody really becomes infected. That doesn't mean it's completely gone away. There is a chance that it could come back in the future. But because we are vaccinated, it's highly unlikely in countries where vaccinations are high that anyone is going to become vaccinated. Now, when we're looking at vaccinations, we've got this idea of herd immunity. Let's put that down there. So put herd immunity down here. And this is just the idea, if I can just make that a little bit bigger. Herd immunity is just the idea that if enough people are vaccinated, it's unlikely that anyone's going to become infected, which means it's unlikely anyone is going to be carrying a disease to then spread it further to more people. And one of the things we're hoping is that with the coronavirus actually is, as enough people become infected, and there are talks that some countries have done this, herd immunity will mean it will stop spreading. But that's only effective if the disease itself doesn't mutate. And already we're hearing reports that there are quite a few different variants of the of COVID-19, which means actually you would have to become immune to multiple types in order for this to actually happen, in order for this to occur. So it's just worth being aware of with that one. But you should be able to talk about vaccinations, and you should be able to talk about how vaccinations work, how they lead to immunity, and actually why they are so important. There are still people out there, there's always conspiracy theorists out there, which will try to indicate that vaccinations are bad. But actually, when you look at the good they have done, I think there is something like a 99.99% chance that vaccinations will be safe. So we've got like 0.001% chance there is something else going on. The alternative is somebody going down with a disease like measles or with some of the other really bad disease, and we can risk death, brain damage, and other issues as well. So actually, the use of vaccinations far outweighs any of the potential risks in any way, shape or form with that. Right, I'm going to leave it there, and I'm going to go on to Google Classroom, partly because my pen's not working because I dropped it in my coffee, and partly because actually that brings us to a natural end point for this particular topic. I will put the link into Google Classroom in a few minutes, and if anyone's got any questions, jump on board, ask some questions, say hello. Again, if we can try and build up some of those numbers. Next session, what we're going to have a look at is going on to how new drugs are developed, some of the issues with that. I'm also going to talk a little bit about evolution, although it comes in a later unit. It's quite important to look at how these superbugs like MRSA and how sort of penicillin-resistant gonorrhea actually has come about. And also we can then talk about how different diseases mutate, which means we might lose immunity if we come across a mutated version of it. So we'll talk about that again, and that will give us a chance to recap some of the things we've just been through. Just to say though, go back through the video link I posted at the start. Also go through and make sure that you're happy with the key sort of terms on here. Come on, there we go. Key terms with lymphocytes, antibodies, antitoxins, phagocytes, phagocytosis, lysis. If you're not sure, ask me again. I can go through it later or rewatch the video. Anyway, hope everyone's well, and I'll see you on Google Meet in a few minutes.